Very good. Brilliant. Excellent. And do not worry about the left lower corner with 141. I'm not going to present as many slides. All right. So very good. So I'm going to talk about Cladribine in MS for obvious reasons. I've been speaking about this, I think, last year when the decision was still outstanding for the EML, and this is obviously great news and a good reason to reappraise, as it were, why this has been chosen as one of these three compounds. So here are my disclosures, and I think key disclosure is right at the top, which is that I'm the CI of Chariot MS, it's on my T-shirt, a trial in more advanced disease using Cladribine as a compound, which perhaps reflects really a little bit the confidence in this drug in terms of safety, but also the long-term efficacy of it. But there's other functions as well, of course. I'm just going to delve into this for a moment to explain what we're doing there, since we're looking at access for disease-modifying treatment. There's obviously more to marginalized groups than only what we're focusing here mainly, which is low and middle income countries, or rather kind of the global movement. But advanced MS is another population that's pretty marginalized. And when you look at it, at least in the UK, they reflect about 35% of the total population who has this type of EDSS, where patients are excluded from DMT and including trials. So we're planning to change that. This is Christine Chapman, who has been very instrumental as a PPIE member of the team to drive this. And this is a simple, quite straightforward two-arm trial that we're currently undertaking, and 200 participants with an EDSS of 6.5 to 8.5. So these are people in wheelchairs, where we're looking at upper limb deterioration as the primary outcome. And that is obviously a relative novelty. And the other one is that there's no upper age limit. So we're really trying to push the envelope quite a bit. This is our first patient who's actually completed the study by now after 2 years, a painter, 70 years old, when she was enrolled originally here with her grandchildren, and a painter, obviously keen to preserve upper limb function. And this is patient number 100. So we're just over the halfway mark now with this lady who's joined us recently. The biological underpinning of this is obviously that we feel the evidence is strong enough to say that even though MS is progressive at that stage, that inflammatory activity is still an important driver of that, and we should try and suppress that as good as we can as part of the overall management of patients. So here's just a recent, I could call it gimmick almost, from a company that focuses on this concept of smoldering MS that might drive this. Just an opportunity to thank everyone who is funding this, as there's 5 organizations who are all agreed to jointly support this study, which we're hoping will complete recruitment later next year. There's a lot of other supporters, and if I had asked at the time the MSIF, I'm sure they would be there as well. But obviously, the focus here is very much on the endorsement by the WHO of those 3 compounds, rituximab, cladribine, glatiramer acetate, and the focus here is on cladribine, which is on this list here in terms of benefits, large benefits, particularly in relapsing MS, trivial harms, and I'll come back to that because it's quite remarkable how that picture has changed over 10, 15 years. when it was originally rejected by the EMA and the FDA based on safety grounds, and now it's actually considered as one of the safest compounds in the certainly in the class of high efficacy treatments. So why Cladribine? Okay, and I'll start out with just a few sort of key trial data that really underpin its use. This is from the original Clarity studies with the extension study that indicated this significant effect on relapse rate, but also on EDSS progression. Now, the way the drug is now licensed, the figures look more beneficial. You may be aware that certainly in Europe and also in the US, the compound is licensed as a drug for highly active disease. And when you sort of pick out that portion among patients that were in studies, then you find that it's near 70% reduction in annualized relapse rate and near over 80% reduction in confirmed EDSS, that's 6 months confirmed EDSS. So it is very much in the level of the monoclonals in terms of efficacy. This is also true when you look at a more recent measurement, so the NIDA rates among the compounds, and here's the OPERA 1 and 2 studies of ocrelizumab with 48%, and here's 47 of the Clarity study. And I always find it quite interesting that the KRMS 1 and 2 studies are actually a bit lower down there, which are obviously the trials of alemtuzumab in MS. Maybe that had to do with the populations that were tested. So it's always difficult, of course, to compare studies, particularly when they're done at different time points as well. Now, Mavenclad, as Cladribine is now called in the real world, also has a positive impact on quality of life. We analyzed this data that Merck had essentially lying around and probably wouldn't have published had we not sort of retrieved them from the EMA. And you can see that quality of life is essentially maintained in those who are on the compound whilst it appears to go down in those on placebo. It's one of the relatively few direct pieces of evidence on terms of quality of life that are out there for disease-modifying treatments. I'm just going to jump over this. I wanted to go into the mechanism of action now, and that is really very much sort of driven, as we've I think, by its impact on B cells. You know that most of the highly effective drugs have very strong impact on B cells, and we have done further research to underpin that it is actually a subpopulation of B cells that you need to suppress. That's the memory B cell proportion. This is the overview of what happens once you give one course of Cladribine. So this is the impact on T cells, so cells of the adaptive immunity, T cells and B cells. You can see here that the overall cell counts obviously drop, but T cells, so here CD4 and CD8, they drop by about half, and then B cells drop altogether, but the memory cells stay down there flat on the floor, as it were, whilst the immature and maturing B cells recover, which is obviously of potential benefit. And so far the evidence seems to support that, that for vaccine response, for example, this is good, whilst for the inhibition and control of MS, this is good as well. The effect on memory B cells is not unique to Cladribine. It is really something that you find in all the effective disease-modifying treatments. There are exceptions. One exception, really, which is natalizumab, which is only to do with the fact that it actually blocks access to the brain. So it's really the mechanism of action, where the others are all systemically working drugs, where the effect on B cells in the entire body, including the brain, is key. And we also have a hierarchy here of response. So when you look at to what degree these drugs deplete and long-term deplete memory B cells, then you find that the highly active ones do that best. This is something that we've also translated into language that we feel is very well understood by patients. So here's some videos that you can access. There's a little series, including one that focuses on immune reconstitution. And since Merck supported the production of these videos, so we advised on their content. I mean, it's easy to find out that this is actually about Cladribine down here, but the others are pretty generic about MS as a disease. Now, just taking one brief step back, and I'm not going to bore you long here, but I always find it useful to know how Cladribine works very generally. So it's a chlorine substituted deoxyadenosine. And this is really what makes it resistant against the enzyme that catabolizes it. Otherwise, it degrades it. And so so this is the normal route of the adenosine deaminase, so through to the purine salvage pathway, and it is in order to actually activate, it needs to add phosphates, or mono-D and then triphosphates, and then it gets incorporated into the nucleus. And there's an antagonizing or a counteracting enzyme, which is the 5-nucleotidase, but it is very much the oxycytidine kinase, this enzyme that is so highly expressed in cells of the adaptive immune system that really drives the effect of the compound. So when you block the catabolism, so the adenosine deaminase, then I mean this obviously accumulates, right? So you get massive accumulation of that small molecule in the cell, which then leads to apoptosis. So this is how Cladribine works, as opposed to the monoclonals that lead to a cell lysis. This is an apoptotic pathway, which explains why it's a little bit slower in the depletion, and also explains why you get fewer side effects acutely with it, because it doesn't knock out all cells immediately. It's a slower process, and therefore you don't get this immediate release across all B cells, say, that lead to rash and other side effects. So here's the distribution of this enzyme that I just mentioned, DCK, across the cells of the adaptive immunity and also non-adaptive. And you can see that there's most of them are in B cells, and there's fewer in T cells and very few down here. And this is how we really explain where the effect is. We know it's clinically very effective. So the fact that it only is a bit mediocre when it comes to T cell depletion, it may play a role here, but we think the key is really the B cell component. Here's just an example of the evidence that really supports the safety aspect in context of antibody generation following vaccination for COVID-19. Here's Cladribine. So this is a so-called violin plot, and I've said I'd consider this: if you have a violin that looks like a cello sitting sort of flat on the floor, you have a problem. And this is obviously true for fingolimod and also for the B cell depleters, because they don't, the antibody generation is obviously a bit inhibited there. So Cladribine is right here, break for infections after vaccination. These are the platform drugs plus natalizumab, and you find that ocrelizumab, and interestingly, rituximab is a little bit of an outlier here, but ocrelizumab certainly one where you find more breakthrough. More on safety. This is obviously a company slide, but I think it's really reflects the data that the main infectious problem with Mavenclad is herpes zoster, so shingles, which is more common. You can see here the figures down here, 0.6 additional cases per 100 patient years compared to placebo. And there's no PML, okay, with Mavenclad, which again, I think speaks to its mechanism of action. Just going to skip this. Moving on to cancer. We're talking about risk, and it's hard to believe that the cancer risk was actually something that led or was part of the rejection. What we did then quite a number of years ago now is essentially comparing the risk difference across the drugs that at the time were actually available. And you can see here that Cladribine sits right in the middle among the compounds. And I always find it interesting that you have at the top a fingolimod study and at the bottom as well, which is obviously a reflection on the fact that over a short term, it's a bit difficult to make that call, but certainly the driver for Cladribine was simply that in the original studies, there were 0 cancers in the placebo arm, whilst all the other trials actually had cancers also in the placebo arm. So long term, I said, so 10 years, this is a comparison with, with databases about malignancy risk, and you can see that the standardized incidence ratio is no different, right, between those observed and the expected malignancy events. There's no hepatological malignancies. There's also no specific group of malignancies that has emerged. I think truly that the malignancy risk is pretty much along similar to what you find in the background population. Since I overheard Therese talking about instruction and teaching education for patients, I thought it'd be useful just to put this up here. Obviously, Mavenclad, or the oral version, has a relatively easy schedule when it comes to testing prior to treatment initiation, and then the 2 blood tests at month 3 and month 7. Even those, I think, are not absolutely necessary because there's very little that you can actually do about them. You can just essentially wait and see. What obviously does sometimes, and that's really quite rare occasions now, happens is that you would consider antiviral therapy, because we know that the significance of the depth of the depletion impacts on the risk of shingles or other herpes diseases. So you could take that as the only reason. But overall, you could probably get away even without doing these tests. So Cladribine is effective in relapsing disease. There is phase 2 evidence that it's also effective in progressive disease. I didn't show the data of that. It's a bit older, from the 90s. It's certainly convenient as an immune reconstitution therapy with rapid immune elimination. Obviously, drug-free pregnancy once you've done the treatment. CNS penetrance, which is now very much highlighted with BTK inhibitors, for example. That is something that Cladribine does as well. About 25% of it gets into the brain, where it can directly attack cells. And by all intents and purposes, it is as safe as any of the disease-modifying treatments. I just want to go and take one brief step back, because we know Mavenclad was licensed in August 2017 by the EMA, and early 2019 in the US. But we've collected information based on the data that we derive from the EMA directly, but also from the cohort that we built using an off-label compound of or preparation of Cladribine since 2014. And this is one of the early things that we did, a video that essentially addresses head-on the issue of risk. I didn't include a QR code here, but you can relatively easily find that if you wish. This is a brief history of how we dealt with the compound, as it were. As many of you will know, my colleague Gavin Giovannoni, he was involved in the phase 3 program, the Clarity trial, but then that was rejected. So what we did then was first search for people who are actually keen to pick up the pieces and try and get the compound to patients, because we thought at the time the data was so strong that it would be an absolute waste to sort of throw it away. And my colleague David Baker called it the search for the white knight, to and used that on the blog for some time to as the figure, but it eventually turned out he would find somebody very close to him. And we've been sharing an office for quite a number of years now. So we had meetings with the UK regulators to establish, do they accept that there is a bioequivalence between the off-label preparation that we were planning to use and the the tested product in the trials, which is obviously the tablet version. The off-label versions are all injectable, either IV or subcutaneous, and they agreed. So that was a meeting in April 2013, so 10 years ago, where that was established. But obviously you need to adjust the dose because the injectable versions have 100% bioavailability versus the about 40+% of the oral version. Got all the data. We had first application to test Cladribine against alemtuzumab, which was not supported. We then applied for a separate scheme to do a trial in Mexico against the standard of care, which would have been Copaxone at the time, which wasn't supported. And but eventually we got through to the Chariot study that is now underway. So this is kind of one outcome of this whole sort of journey, this process, and alongside, I think what we've done to underpin the use in less fortunate or less wealthy countries. There's a number of papers that were produced at the time, and this is obviously all out there. Now, the MSIF and Joanna, the team, they've produced a great set of slides. There's some of them just to really illustrate what the barriers are, and you all obviously know that. I think one thing to say is also that when we're looking at repurposing or off-label use of drugs, that most of the compounds that have been developed to this day is that has been developed from an off-label to an on-label version by big pharma. Okay, and when you look at it here, for example, Cladribine is obviously a compound that's been around since the early 90s for hairy cell leukemia, and then became initially Movectro, and then now Mavenclad. Fingolimod was developed for a different indication, and is essentially just reused. Leflunomide became teriflunomide, and a very expensive active metabolite of leflunomide, and then thereby Orbaggio. Campath, we know the story, all that it was just reformulated, repackaged, smaller dose, much more expensive. So this is what you get through the process of trials, because they are obviously a huge investment. So I'm not only kind of, although it sounds a bit like, perhaps I'm not kind of dismissive and critical. It's just what happens on the ground when these compounds are being developed. And it obviously applies to an extent also to rituximab, and then ocrelizumab and ofatumumab and obinutuzumab now. I've gone back to one of the overviews here, which you can get out of Joanna's and colleagues' paper, and just looked at the principles. And essentially was reflecting on how we would have done, or how we did, as it were, on these various accounts when we use compounds such as Cladribine off-label. It's obviously driven by the need for people. Should be evidence based. There should be shared decision making, appropriate information, and outcomes should be carefully monitored. And I think we've actually ticked quite a number of these boxes. So here's the information that is publicly available and that we made publicly available back in the day when we started using the compound. Is available through SlideShare for patients. And this is the report of the first 208 patients. We had a previous paper out on us. You can see how well embedded that was, certainly in the UK MS community. So this was nearly 40 authors here who all kind of either referred or contributed otherwise to this. Obviously, what we had to do was, we called this, by the way, treating the ineligible case, because there were restrictions. Certainly in 2014, there were quite a number of the drugs that we have now, obviously not around. So we called that treating the ineligible because of the strict NICE criteria for disease modification. So we essentially adapted our treatment using the injectable compound to what is happening now with the tablet. Just slightly altered it, and that was really against the backdrop of the concern that was there still about lymphopenia and a little bit about obviously cancer, but mainly really related to lymphopenia. So we adapted the dose individually to lymphocyte count, and we did that for treatment course 1 and treatment course 2. Sometimes when people arrived with a lymphocyte count below 1, we actually didn't give them further treatment. Below 0.5, we didn't give them any further treatment at all. And you'll find that I didn't bring the slide with that, but you will find that the second course, only about a third of patients had the full second dose at that point. And that is relevant in a moment when I show you something about the longer term follow-up of these patients. These were baseline demographics. The median EDSS was 5.5, so illustrating that this is obviously a cohort that is skewed towards those with more disability, rather more progressive disease. At the time, nothing was available, and we did treat people based on disease activity, either MRI or elevated neurofilament levels in the spinal fluid. This was the overall group. There's a bit too small, but I'll obviously share the slides afterwards. These were the 208 patients we treated. Adverse events, I think, again, all a bit too small, but very much along the lines of what we knew from the clinical program, the Clarity program. We got some really, I think, good supportive evidence from doing spinal taps. About 10% of patients had neurofilaments tested, and you can see that they virtually all dropped massively. And this is a logarithmic scale, so you can see how CSF neurofilament, which is obviously a marker in MS for inflammation-driven neurodegeneration, how that sort of drops down. And the blue dots are all kind of healthy levels, and the red ones are all not healthy. So it does it really well. Here are the data for NIDA, knee pads, so there's no evidence of progression or active disease. So this is 60% here. If you remember from the ocrelizumab study in primary progressive MS, this was just around 30%. But we're obviously mindful that these are small numbers. This is more audit type of data. This is more in support of doing further research rather than giving the final answer, as it were. We've now, and this is just actually a couple of weeks ago, been accepted, published some more data on this cohort, which has now grown to 264. I mean, in reality, we are close to 300 now. and that's been accepted by TAND. So here we looked really at how long or how many will develop new disease activity. This is something that is obviously around as a big question, because currently the horizon is, okay, 4 years, 2 courses that should last for about 4 years, and then we don't know exactly what to do. So we looked at them over 4.5 years on average. Okay, this was up to 7.5 years actually. But you could see that over nearly 180 patients out of these 264 had no further disease activity based on MRI or relapses, and had no further immunotherapy. And this is the population of interest here: 13 who had no further immunotherapy, 22 who had new disease activity and got another course of Cladribine, and then another 22 who had disease activity and went on to a different immunotherapy. So we were really interested in the factors that might predict that patients develop new disease activity. This is a lovely Sankey that our statistician produced just to illustrate that here you've got 68% who had no further immunotherapy after that. And you can see what patients received then. Some switched to ocrelizumab, here rituximab, alemtuzumab. Here we had half of those on Cladribine went on to Cladribine tablets, and the others, because they were not eligible for Cladribine tablets, went into subcut treatment. Some did never have a second course. And this is just the kind of real world data that you get in your clinic, right? So one went on to HSUT. And this is really I think the key outcome to look at when in relation to when is there new disease activity. That's 236 patients. So we took those out that didn't have a second course. And you can see that any new MRI activity at baseline is actually predictive, a risk factor for new disease activity after an average of 4.5 years. You can see here the hazard ratio is about doubled at that point. And the dose, I referred to the fact that we reduced the dose according to individual lymphocyte count, also impacted on this. It's a slightly counterintuitive because it's 0.84, but it is because with smaller with a higher dose, we had a lower risk. So a smaller dose means higher risk. Just a final word on the resource poor setting. This is from 2018. So we were obviously at that point very mindful, and this is very much what underpinned our whole program in that respect. Could this be something that could be of use for people in settings where there's not healthcare not so well supported? And when you look at the prices here, so this is Loistatine, which is an IV product. This UK price, 1,330. So this is for a complete treatment course. Okay, so do we have actually Cladribine tablets somewhere here? I'm not 100% sure. But certainly when you look at natalizumab and Lemtrada, and here it is right above. So this is what's there. And nevertheless, remember, it's considered to be very likely cost effective simply because of the longevity of the effect. So really to conclude, I'd say that both on and off label Cladribine appears safe, effective, and apparently affordable. There's no one size fits all, of course. And my question really in the context of talking about off-label Cladribine is, now that Mavenclad is on the AML list, what should we actually do about off-label Cladribine and perhaps more generally off-label drugs, bearing in mind obviously that rituximab is off-label use as well. So thanks very much. It's obviously a big team effort. This is a smaller part on 11D, our daycare unit, and lots of people involved along the way. And we were all much younger when it started. Thanks very much.